Good morning, learners. Welcome. Today, in this session, you will be studying the biomolecules, the carbohydrates. In due course of time, you will understand the concept of biomolecules and the basic principles of chemistry and its importance in the biological systems. At the end of the session, of this module, you will be able to appreciate the occurrence of carbohydrates, various types of carbohydrates, its stereochemistry, and its significance in the biological systems. Why carbohydrates? These carbohydrates principally act as fuels and give energy as they get degraded. They are the major structural framework, form the major structural framework of both the DNA and the RNA. They also form the skeletal elements of the cell wall of the bacteria and the cellulose. As you know, the bacterial cell wall is NM and NAG, and the plant cell wall is made up of the cellulose. These are all the important components of the carbohydrates. And the exoskeleton of the arthropods, if you can recall the cockroaches, they have a thick exoskeleton. This also attributes or is an important component of the carbohydrates. They also play a major role in the cell to cell recognition, and these carbohydrates are also linked to many proteins and lipids. If you can recall the fluid mosaic model, wherein it is a carbohydrate which is attached to both the lipids as well as the protein subunits. These carbohydrates are found abundantly in nature, and they are in the form of hydroxy alcohols, and therefore they are referred to as the polyhydroxy compounds. Either they have the aldehydic group or a ketonic group. They end with the suffix OSE. The carbohydrates can be broadly categorized into the monosaccharides. Mono is single, saccharide is sugar unit. That is, on hydrolysis, breakdown, degradation, they yield a single monosaccharide, that is, a single unit of sugar. Then it is referred to as the monosaccharide. They could be also placed under oligosaccharides. Oligo is when they yield somewhere between 2 to 10 monosaccharide units. Then it is called as an oligosaccharide. Then you have the polysaccharide, many saccharide sugar units. That is, when the carbohydrates, when they are broken down, degraded, or cleaved, they yield innumerable, indefinite, infinite, a number of polysaccharide units. And therefore, since they give you a number of sugar units which are innumerable, undefined, infinite, indefinite sugar units, it is referred to as the polysaccharide. The carbohydrates principally contain the glycerol, which is considered as the parent compound. Though this glycerol is not a carbohydrate, it is still considered to be a parent compound. Why? Because it could be oxidized to give you an aldehyde, which is called as the glyceraldehyde, or it could give you the ketone, which is dihydroxyacetone. So these form the basis for all the carbohydrates. Based on the functional group it contains, If it contains the CHO group, which is an aldehydic group, glycerol is referred to as glyceraldehyde. So this glyceraldehyde contains 3 carbons in it. So wow, what is it called? It is called as a triose. Why triose? Because it contains 3 carbons in it. It could be referred to as a tetrose. Why tetrose? If it contains 4 carbons in it. That you shall just see in subsequent slides. What is a tetrose? Then you have a pentose. Why pentose? Because it contains 5 carbons in it. If the molecule contains 5 carbons, it is called as a pentose. All right. So based on the number of the carbons, it could be a triose, a tetrose, a pentose, or a hexose. So what does triose mean? Triose means since glyceraldehyde contains 3 carbons in it, it is called as a triose. Why aldotriose? Because it contains an aldehydic group. Why glycerol? Because the parent compound is the glycerol, which is an alcohol. If you recall, as I had written in the earlier slide, it is a glycerol. This glycerol is OH, is replaced by CHO, which is an aldehydic group. So therefore, it is a aldotriose. On the other hand, the dihydroxyacetone. What is acetone? It is an ketonic group. This ketonic group is present in the second carbon. So, how many carbons in total? 3 carbons, 1, 2 and 3 carbons. So, it is a triose. And dihydroxy. Why dihydroxy? Because 2 alcoholic groups here, hydroxyl groups here. So since it contains 2 OH groups in it with a ketonic group, it is called as a dihydroxyacetone, which is a ketotriose. Keto because it contains a ketonic group. Triose because it contains the 3 carbons in them. Now you have learnt what a triose sugar and aldose sugar means and what a keto sugar means. Now we come to the next aspect. The carbohydrates can exist as isomers. Now what is an isomer? Iso is same, mer is molecule. Same molecule with a structural formula being different. It has the same molecular formula but a different structural formula. Alright. So look at this structure. This is a glyceraldehyde. So what type? It is a triose. It is a triose. 1, 2, 3 carbons. So triose. Aldehydic group. So it is a ketose. Is it a ketose? No. It is an aldose. It is an aldose sugar. Now, I have marked this carbon with a star. Look at the second carbon here. I have marked a star there. Now, what does this indicate? This indicates that it is asymmetric. Now, what is asymmetric carbon? This is a chiral carbon. Okay. So, look at the numbering there. This would be number 1. This is number 1, number 2 and number 3. The carbon 2 is a chiral center of glyceraldehyde. Therefore, it can exist in 2 forms. That is the L form and the T form. By convention, sugars are written in such a way, in such a manner, that most oxidized carbon is at the top. Which is the most oxidized carbon? The most oxidized carbon is at the top, that is the CHO group. the chiral center away from the most oxidized carbon is used for determining whether it is the D or the L form. Now look at the OH group. The OH group in L-aldehyde is towards the left. Since the OH group is towards the left, it is with a L configuration. And if the OH group is towards the right, then it is the D configuration. Did you understand, students? So this is the most oxidized group. The asymmetric carbon away from the most oxidized group is the chiral center. Now this chiral center is used for determining whether the molecule is the L or the D form. If the OH group is towards the left, then it is the L form. If the OH group is towards the right, then it is the D form. Look at these aldoses. I was just speaking to you about the triosis all this time because it contained 3 carbons in them. These are the tetroses. Why tetroses? 1, 2, 3, 4. 4 carbons, 1, 2, 3 and 4. Here again just see 1, 2, 3 and 4. Which is the most oxidized form? This is the most oxidized form. What type is it? It is a CHO group. So what is it? It is an aldose. So it is an aldose sugar. Further, it is an aldotetrose. Why? Because it contains 1, 2, 3 and 4 carbons in it. So therefore, it is an aldotetrose. Now you have learnt what a aldotriose is, what a aldotetrose is, what is what a ketotriose is. Now just see this. How many chiral compounds? 1, 2 and 3. Now this is the farthest away from the most oxidized carbon. This is the carbon which is farthest away from the most oxidized carbon. And look at the position of the OH. The position of OH is towards the left. So therefore it is the L-erythrose. Now just look at this. Where is the position of the carbon, the OH here? The position of the OH is towards the right. Alright, so what type of the sugar is this? How do you name it? This is D-erythrose. Why D-erythrose? This is the farthest or the most, the carbon which is away from the aldehydic group. Since this is farthest away from the aldehydic group and the position of the OH is towards the right, this is called as the D-erythrose. Come to the other molecule, which is also an aldotetrose. Look at the position of the OH from the most oxidized carbon, the CHO. It is towards the left, and since it is towards the left, it is called as the L-triose. And since the OH is placed towards the right in the farthest carbon from the most oxidized carbon in this group, it is called as the D-triose. Learners, now you are familiar with the L and the D type of the aldoses. Now come to the ketosis. What type of this is a ketotriose. Why ketotriose? 3 molecules, there are 3 carbons in it, and the second carbon contains a ketonic group, so therefore it is a keto sugar. Further, it is a triose, keto triose, because it contains the functional group ketonic group and 3 carbons, therefore keto triose. Now look at this, it contains 4 carbons in it, therefore it is a tetrose. Further, it contains a ketonic group in it, so it is a ketotetrose. Look at the OH position. The position of the OH is towards the right. Now, which is the most oxidized carbon in this case? The most oxidized carbon is this, and the farthest away carbon from the most oxidized carbon is OH is placed towards the right. Therefore, it is a erythrulose. So, had it been a aldose, then you would have written erythrose. Otherwise, it is given as erythrulose. The different important monosaccharides, what you come across is the D-glyceraldehyde, which is the simplest sugar. D-glucose, it is the most important in the diet. And you have the D-fructose, which is the sweetest of all the sugars present in the fruits. When you eat the fruits, they are very sweet. Why is it? It is because of the presence of the D-fructose. Then you have the D-galactose, the carbohydrate or the monosaccharide or the sugar that is present in the milk, and the D-ribose, which is present in the RNA, which is the genetic material. The epimers. The epimers are nothing but those carbohydrates that differ in their stereochemistry at only one position. Alright, so those molecules which differ in only one position is referred to as the epimers. Just look at this molecule, you are familiar with this, it is the D-glucose. You start numbering, you must be very familiar with the numbering learners. The most oxidized carbon, you will number it as number 1. And as you go down, you will start numbering it as the farther the carbons, you will start numbering it as 1, 2, 3, 4, 5 and 6. So, which is the most chiral compound now? The most chiral compound is the C5. Why? Because this is the farthest carbon, which is called as the chiral center. And since at the chiral center, the OH is towards the right, it is called as the D-glucose. Now compare this with the other molecule, the D-mannose. As I told you, you will start numbering the most oxidized carbon as number 1, then number 2, 3, 4, 5 and 6. Now, just look at the carbon 2. In the carbon 2, compare it with glucose. At carbon 2, the presence of OH is towards the right in case of glucose, but at the position 2 of the mannose, the OH is present towards the left. Now compare carbon 3, 4, 5 and 6. The rest all are the same. Now there is a difference in the arrangement of the OH at only one point, which is the second carbon. And this is the epimer. So what does it mean? Look at the orientation. In the right, you have the OH at the right in glucose, and towards the left in the mannose. The rest are all the same. Only change is at C2. Difference is only at one position. So therefore, mannose is an epimer of D-glucose. Did you understand? The second epimer, the D-galactose, you start numbering same as in the earlier case. The most oxidized carbon, 1, that of the aldehyde, 2, 3, 4, 5 and 6. Now look at the position of 4. In case of the galactose, at the 4th carbon, OH is present towards the left, whereas at the 4th position in the glucose, the 4th position carbon, OH is present towards the right. The rest all, 1, 2, 3, 5 and 6 remain the same in both the molecules. Now change is seen only at the 4th carbon. What does it indicate? It indicates that the galactose is an epimer of the glucose at position 4. Hope it is clear, learners. So what are epimers? Epimers are those compounds or those carbohydrates or those sugar molecules that differ only in their stereochemistry at one position of the molecule. Then they are called as the epimers. What type of structure is this? This is an open chain structure. Since it is in linear form, since it is linearly arranged, you call this as the open chain structure. These open chain structure can undergo cyclization. What is cyclization? They can undergo a ring formation. Now look at carefully the glucose, carbon 1, 2, 3, 4, 5 and 6. Now it is the cyclization that is formed between the carbon 1, that is the CHO group, and the carbon 5. It is through the hydroxylic group. So, it is through the hydroxylic group of carbon 5 and the CHO group of the carbon 1 that you see the cyclization. So what is cyclization? It is nothing but the formation of a ring structure. So it is formed between the OH of the 5th carbon and the aldehyde to form a hemiacetyl, wherein the OH of the 5th carbon and the oxygen from here comes, takes up the proton, going to form a 6-member ring with CH2OH, which is at the carbon 6 being intact. So what is cyclization? It is the process wherein there is a ring formation between the hydroxylic group or the alcoholic group of the carbon 5 with that of the aldehydic group of the carbon 1. So the carbon 1, 2, start numbering from the most oxidized carbon. So carbon 1, 2, 3, 4, 5. And CH2OH, look at this, it is intact in both the structures, and therefore the formation of a 6-carbon ring structure is obtained through the process of cyclization. Now, following the cyclization, there is the formation of a ring structure, and when it is a 6-carbon or the 6 carbon structured ring, then it is called as a pyron, and a 5-carbon structured ring, then it is called as a furon. So these molecules will now, by which you could be able to give the pyron name as well as the furon name. So a 6-membered pyron is referred to as the pyronoside, example glucose. A 5-membered ring structure called as the furon is referred to as a furonoside, example fructose. Further, they can be either the alpha or the beta. Alright, now look at this. It contains a 6-membered ring. CH2OH is the 6, 1, 2, 3, 4, 5 and 6. Okay, 6-membered ring. Further, they are nothing but the different stereoisomers. They are referred to as the alpha and the beta. Because when it is alpha, it is in a transposition. Look at the OH. It is in a transposition to that of the CH2OH. Alright, and if it is beta, it is in the cis position to that of the CH2OH. So when it is in the trans position, then it is the alpha, and when it is in the cis position, it is the beta. Mutarotation. Rotation, what does it mean? It will shift, it will rotate what? The light of polarization, the plane of the light of polarization. Excuse me. In aqueous solutions, these anomers have the ability to interconvert the open chain structure, alright, and this is the transient formation of the open chain structure, and this transient process is what you call as the mutarotation. So once there is the mutarotation, you give the degree of the light of polarization to whether it rotates to the right or whether it rotates to the left, and therefore you denote it as the small d and the small l form. The small d will rotate the light of polarization. These molecules will rotate the light of polarization towards the right, then it is the small d form, optically active. The other type is the L form, which is called as the levorotatory. When they are able to rotate the light of polarization towards the left, then they are said to be levorotatory. When it comes to mutarotation, you will be writing whether they are dextrorotatory or whether they are levorotatory. Dextrorotatory, if they are able to rotate the light of polarization towards the right, then it is indicated at small d and it is written as dextrorotatory. If they are able to rotate the light of polarization towards the left, they are referred to as the levorotatory and given as small l. Coming to the properties of aldehydes, they have the ability to undergo the oxidation. The sugars, those which contain the aldose CHO group, they have the ability to undergo the process of oxidation because the aldoses act as the reducing agents. Any sugar with a free aldehydic group is said to be a reducing sugar. Like for example, the glucose. During this process, the glucose is converted into an acid, gluconic acid. Glucose is converted into the gluconic acid. Now, during this process, it is able to reduce the failing solution, that is the Benedict solution or whatever Benedict solution or the failing solution, into the blue copper 2 to copper 1. That is the red precipitate of copper oxide is an indication that it is a reducing sugar. The OH group of the monosaccharides can also be readily converted into the esters or the ethers, the esterification done by an acid chloride. There is also the formation of the glycosidic linkage. What is the glycoside? So, when two molecules of the monosaccharide units or the sugar units, in case of the oligosaccharides and polysaccharides, they join together, conjugate together, aggregate together, linked together by a linkage, which is called as the glycosidic linkage. Now, what is this glycosidic linkage? This is one glucose molecule. Just look at this glucose molecule. It is a 6-membered ring structure. 1, 2, 3, 4, 5, 6. 6-membered ring structure. And this is, where does this come from? This is from the, as I told you, during the cyclization. The OH group of the 5th carbon is cyclizing with the CHO group of the 1st carbon. You remember? Yes. So, following which there is a closed structure, and this closed structure is called as the ring structure. So, this is the glucose molecule. Now, this is alpha. Why alpha? Because it is in the transposition, as that of the CH2O group. Had it been here in the cis position, then it could be the beta. Alright? Okay. So now, you draw one more glucose molecule here. Both alpha trans glucose, that is the alpha glucose. Now, there is the linkage established between the 1st carbon and the 4th carbon. The 1st and the 4th carbon. Alright? The 1st and the 4th carbon. So, what does it indicate? What does it indicate? 1,4 linkage. Look at this. Since carbon 1 of 1st glucose is involved in the bond formation with the carbon 4 of the adjoining glucose. It is what type of linkage? 1,4 linkage. Further, how do you denote whether it is an alpha or a beta 1,4 linkage? Further, you denote it whether it is an alpha or a beta linkage. Look at the position of the OH. The OH of the first glucose is in the trans position. So since it is in the trans position, it is said to be alpha. What type of linkage is it now? It is alpha 1,4 linkage. Why alpha 1,4 linkage? Alpha because the OH is in the trans position or in the what you call as the alpha position. So the OH, since the alpha is or the OH is involved in the bond formation in between the first and the fourth carbon, it is called as the alpha 1,4 linkage. And this type of a linkage which is established between the two adjoining the glucose molecules or two sugar units is called as the glycosidic linkage. So what is glycosidic linkage, learners? It is nothing but a linkage that is established between one monosaccharide unit with the adjacent monosaccharide unit through the formation of a special type of a linkage is called as the glycosidic linkage. Further, it can be alpha 1,4 linkage as you see in case of maltose, or beta 1,4 linkage. It could be any type. Alpha 1,4 linkage, why? Alpha because the OH is in the trans position, therefore it is alpha. 1,4 because the first carbon, the first carbon is forming a link between the fourth carbon. So therefore it is the 1,4 linkage. Further, these oligosaccharides can be either categorized into the disaccharides or the trisaccharides or the tetrasaccharides or the pentasaccharides. Disaccharides, why disaccharides? Di, 2, saccharide sugar units. The sugar units on hydrolysis, these sugar units will yield 2 monosaccharide units, as in case of sucrose. In case of sucrose, you get, when they are broken down, 2 monosaccharide units, 1 plus 1, glucose plus fructose. The glucose is 1 monosaccharide unit, fructose another monosaccharide unit. So therefore they are called as the disaccharides. Trisaccharides, here 3 monosaccharide units, they are joined together by the glycosidic linkage to give you 3 monosaccharide units. The tetrasaccharides are nothing but 4 monosaccharide units which are joined together to give you one complex large molecule. Then you have the pentasaccharide units, the same. The polysaccharides. These polysaccharides are nothing but they are joined together, conjugated together, attached together by 10 or more monosaccharide units with the glycosidic linkages. They can either be the homopolysaccharides. Homo is the same. Polysaccharides That is, they are composed of the same monosaccharide units. So number of innumerable, indefinite number of monosaccharide units join together, conjugate together through the glycosidic linkages to give a large macromolecule. And this large macromolecule composed of same type of monosaccharide units is called as the starch or the cellobiose, for example. They are composed of the same monosaccharide units. The heteropolysaccharides are nothing but those monosaccharide units which are different from each other. They conjugate together, attach together through the glycosidic linkage to give a large macromolecule. For example, the hyaluronic acid, chondroitin sulfates. Further, these polysaccharides can also be called the digestible or the non-digestible polysaccharides. Why digestible? These polysaccharides, when consumed, can be easily broken down. For example, the starch. You consume every day rice, chapati, dosa. What does it contain? It contains principally the starch. As you consume these foods, due to the process of the grinding, there is the action of the amylase, which is an important component of the enzyme, the saliva. It gets partially digested in the mouth itself. Following its swallowing, what happens in the stomach? Again, it is acted on by a number of enzymes, the pepsin, renin, lipases, all these. Again, it gets digested and part of it is utilized as a source of energy and part of it goes for the storage. Whereas in man, you eat a lot of fruits and vegetables. They contain cellulose in them, which is an important component of the cell wall of the plant. The cellulose, what the human being consumes, is not digested. It is instead given out, excreted out, thrown out as roughage, and this prevents constipation. Why is it called non-digestible? Because the human beings lack the enzyme cellulase, therefore does not get digested and is thrown out of the body. So this polysaccharide is referred to as the non-digestible polysaccharide. As I told you, it is an important component of the storage. It could be in the form of, as you have seen in the plants, it is stored in the form of fruits and vegetables. Alright? And then you have the structure, they also form the important structural components. Like for example, in case of the exoskeleton of the arthropods, at the beginning I told you, the cockroaches, they have a thick exoprotective skeleton. So this is made up of the carbohydrate. Coming to the biological importance, they are important sources of energy, they act as precursors, then the components of the cell structures, and also form the constituents of the nucleic acid, coenzymes, and the flavoproteins. They also serve as lubricants in case of the joints of the skeletal muscles, both in the humans as well as the animals. A quick recapitulation of what all you have studied today, learners. You studied what a monosaccharide is, the importance of the monosaccharides, the reactions involving the monosaccharides, then what are oligosaccharides and the polysaccharides. You learned what a glycosidic linkage is, how it establishes itself to give you the oligo and the polysaccharides, and what the reducing and the non-reducing sugars are, what aldoses and ketoses are. So, thank you students. Wishing you all the very best.